Our bodies are comprised of trillions of cells. Each cell plays its specific role in maintaining homeostasis and keeping the entire organism alive and functioning properly. One of the fundamental and most important roles of cells in maintaining health is to be able to regulate what materials can pass into the cells and what can leave. This function is germane for keeping the entire body alive and underlies major processes like maintaining blood pressure, digestion, electrolyte balance, and so on. Transport and permeability are carried out by the plasma membrane. In this unit, we will define what permeability is and examine ways in which living cells can respond to their internal and external environments by regulating permeability. Let's begin by defining what permeability is. Permeability of a cell membrane is the property that determines what substances can enter or leave the cytoplasm of the cells. In general, there are four subcategories of permeability and two types of membranes, living or non-living. The first subcategory includes impermeable membranes, which means that nothing can pass through the membrane. Second, some types of membranes are freely permeable and permit anything to pass through. Third, semi-permeable membranes are any physical barrier, living or non-living, that are permeable to some molecules but not to others. Permeability properties in this case is by virtue of the size of the pores in the membrane. Semi-permeable membranes are used extensively in research to separate different size molecules, such as proteins, from one another. They are also used in kidney dialysis. The last subcategory, selectively permeable, applies to living membranes that possess the ability to change their permeability properties and can select for or against a substance by opening a channel or activating a transporter. Why is it important for cells to be able to regulate what materials can cross the cell membrane? Our cells are bathed in an extracellular fluid called interstitial fluid that is derived from blood plasma. Interstitial fluid contains thousands of substances, including amino acids, sugars, fatty acids, vitamins, hormones, neurotransmitters, ions, and waste products. We will see in subsequent slides that the composition of substances inside the cell is very different from the composition of substances in the fluid that bathes the cells. And this fluid is different from the blood. Without these differences, cells would cease to function properly. In other words, homeostatic imbalance results if the distribution of ions, molecules, and electrolytes inside and outside the cell are not maintained in composition and concentration within specific limits for normal cellular function. When cells suffer from severe states of fluid and electrolyte imbalance, for example, disease states can develop, system-wide, and this can lead to death of the body. To understand how cells select materials to enter and leave, let's define what body fluid is. Body fluid is an aqueous solution consisting of water, the solvent, in which there are dissolved particles such as ions or molecules. The particles are called solutes. Life on this planet is based around the unique molecular structure of water, and no other fluid can replace water in living cells. Water is called the universal solvent because, given enough time, almost everything dissolves in water. Water prevents rapid swings in temperature, which helps keep body temperature stable. Water has high surface tension or cohesiveness because hydrogen bonds between water molecules hold them together. To test water cohesion, put a drop of water on your fingertip and let it hang there. You are observing hundreds and hundreds of individual water molecules sticking to each other. This phenomenon is also what helps cells maintain their shape and makes blood an excellent transport medium. The solutes that are commonly found in body fluid include ions, electrolytes, and metabolites. Ions are atoms or molecules that carry a charge when dissolved in water. Common ions include Na+, K+, Mg2+, K2+, Cl, PO43, and proteins. Positively charged ions are called cations and negatively charged ions are called anions. Electrolytes are ions that will conduct an electrical current in solution. Sodium and potassium are two very important examples. Molecules that are not charged in solution are called non-electrolytes. Glucose is a very important non-electrolyte for cells. This table shows the main electrolytes found in body fluid. This includes fluid inside and outside the cell, as we shall see. The table above shows you the relative concentration of the major electrolytes found in blood. Note the relatively high concentration of sodium. Also notice that a normal range is given for each. The normal range is the concentration that is optimal for normal function. Values above or below the normal range of each electrolyte indicate some sort of homeostatic imbalance. A simple blood test can tell a physician a lot about the health of the patient. Metabolites are usually small molecules that are intermediates or products of metabolic processes. Glucose is used for ATP production by mitochondria. Urea is a metabolite of protein breakdown, and bilirubin is produced during the breakdown and recycling of red blood cells. Proteins are large molecules that carry a negative charge in solution. They are referred to as colloids. We will see later on that certain proteins in plasma are important for regulating fluid balance and blood pressure. We have been alluding to body fluid. Now let's look at exactly where body fluid is located within the whole organism. The body's fluid is compartmentalized. The compartments include the intracellular within cells fluid or cytosol, and the extracellular fluid, which includes all fluid outside of cells. Surprisingly, the intracellular or cytosolic fluid compartment makes up 62% of our total body fluid. This seems out of proportion because cells are usually microscopic. However, our bodies consist of trillions of individual cells, and collectively, that adds up to about 62% of our total body fluid. The extracellular fluid compartment is divided up into the fluid that bathes our cells, called interstitial fluid, and plasma, which is the liquid portion of whole blood. Each of these fluid compartments may contain similar solutes. However, the concentrations of the solutes are very different when the three compartments are compared. Some solutes are only found in a specific compartment. The intracellular fluid compartment differs significantly in the types and concentrations of ions, electrolytes, molecules, and metabolites compared to the interstitial fluid. Interstitial fluid also contains some molecules not found within cells, including certain sugars, fatty acids, vitamins, hormones, and other metabolites. Similarly, the composition of blood plasma is different from both intracellular and interstitial fluid. To remain healthy, each cell must extract from the mix of molecules and ions that bathe it the exact amounts of substances it needs at specific times. The key to these life-sustaining differences is the plasma membrane, as it is the ultimate regulator of what can enter or leave the cell. Let's review the structure of the plasma membrane, since it is the key to understanding permeability and selectivity of cells. Our current idea of plasma membrane structure is called the fluid mosaic model. The plasma membrane consists of a double layer of lipids with embedded proteins. The lipid bilayer is made up of about 75% phospholipids. Phospholipids are characterized by having a polar head, which means it is water-loving, hydrophilic, and can interact with the water component of body fluids. The other part of the phospholipid molecule consists of two fatty acid tails, which are nonpolar and hydrophobic water-fearing. Notice that the tails of the fatty acids point inward toward each other, whereas the polar heads are facing the watery environment inside and outside the cell. Thus, The plasma membrane effectively separates the intracellular fluid ICF from the extracellular fluid ECF. About 5% of the membrane lipids are glycolipids, which have a carbohydrate group attached to them. Another 20% of the lipids are cholesterol. Cholesterol stiffens plasma membranes, whereas phospholipids make them more fluid. The integral membrane proteins play a very strategic role in selectivity. They are inserted into the bilipid layer, and many go all the way through it from inside to outside. These are called transmembrane proteins. Most of the integral membrane proteins play a role in transport. They can serve as pores or channels, as carriers, receptors, or as enzymes. Peripheral membrane proteins attach loosely to the integral membrane proteins on both the inside and outside of the membrane. The glycoproteins on the extracellular surface help to form a glycocalyx, important for cell recognitions and other functions. The proteins on the intracellular surface are part of a cytoskeleton that helps give support to the cells among other functions. Up to now, we've established that membrane permeability determines precisely which materials can enter or leave the cell, and that living cells are selectively permeable. It is important to keep in mind that our bodies are made up of 200 different kinds of cells. As seen in the diagram, each cell type has a unique structure and function in the body. To effectively carry out their specific roles for the body, each type of cell selects free passage of some materials and restricts the passage of others. What can pass through is determined by membrane properties, like the kinds of lipids and proteins that comprise it. The properties of the solute can also influence membrane permeability. Permeability is affected by size, charge, shape, and lipid solubility of the solute surrounding it. There are two major ways that substances move into or out of the cells through the plasma membrane. In passive transport, solutes are moved across the plasma membrane without the cell having to expend any energy. Diffusion, osmosis, and filtration are examples of passive transport by cells. The second method requires the cell to spend some energy in the form of ATP. This is called active transport because active transport always requires the cells to burn some energy. Remember A for active and A for ATP. There are three different mechanisms occurring at the plasma membrane by which the cell can transport materials. Diffusion is always a passive process. It is based on the random motion kinetic energy of particles, so the cell doesn't have to invest any energy. The second way is called carrier or channel-mediated transport. This mechanism requires help from integral membrane proteins which act as facilitators or channels that help materials get through the membrane that may be too big, or carry a charge, or are hydrophilic, not lipid-soluble. Carrier or channel-mediated transport can be either passive or active depending on what needs to be transported. The third major mechanism is called vesicular transport. In this process, materials are moved into or out of the cell in small membrane-bound packets or vesicles, such as secretory vesicles. This process is always active and requires the cell to expend ATP. Let's look in more detail at each of these processes, beginning with passive processes. What determines whether or not a substance can passively get through the plasma membrane includes the substance's lipid solubility, size, appropriate channels, or carrier proteins. Passive processes include simple diffusion, facilitated diffusion, channel-mediated diffusion, and osmosis. We will begin with simple diffusion, which can be thought of as a particle distribution process. This process is based on the tendency of particles' molecules or ions to scatter evenly throughout the environment in a defined space. The continuous random motion of the particles will result in their becoming evenly distributed throughout that space. The overall effect of this erratic movement is that particles move away from areas where they are in higher concentration to areas where their concentration is lower. We call this behavior diffusion along or down the concentration gradient of the particle. The difference between the high and low concentrations of the particles is called the concentration gradient. How rapidly diffusion occurs depends in part on the size of the particle. Smaller particles diffuse faster, and by temperature. The warmer the surroundings, the faster the particles move. Simple diffusion eliminates the concentration gradient. The particles bounce off of each other until they are evenly distributed within the defined space. When this occurs, we say that equilibrium has been reached. Note, the particles never cease their random motion even after equilibrium has been reached. They just remain evenly distributed. This is an important point to remember. Equilibrium does not mean that diffusion does not continue, only that there is no longer a concentration gradient and hence no net movement. Molecular motion, which underlies diffusional transport, never ceases. Let's look at one example of how diffusion occurs in the body. Carbon dioxide gas, indicated by yellow pentagons, is a waste product of aerobic metabolism. It must be eliminated from the body. You know that when we breathe, we inhale oxygen and exhale carbon dioxide. How does the carbon dioxide generated by our cells get to our lungs so it can be eliminated? The answer: simple diffusion down carbon dioxide's concentration gradient. Because our cells produce carbon dioxide as a waste product, its concentration is highest within the cell. Therefore, carbon dioxide diffuses down its concentration gradient into the interstitial fluid, which has a lower concentration of carbon dioxide than the cell. Blood-carrying capillaries are within a few microns of our cells. Since blood is being moved along through the capillaries, carbon dioxide is being swept away. Therefore, the concentration of carbon dioxide in the capillaries is lower than in the interstitial fluid. Eventually, the blood returns to the heart and is pumped to the lungs where the carbon dioxide gas is eliminated. Oxygen is transported to the cells in a similar way. Oxygen is necessary for the production of ATP by the mitochondria. This illustrates the life-sustaining importance of the passive process of simple diffusion. Several conditions affect diffusion. These include distance, particle size, temperature, gradient size or steepness, lipid solubility, and electrical forces. Distance refers to how far a particle must move to achieve equilibrium. Obviously, the smaller the distance, the quicker the concentration gradient is eliminated. That is why our cells are only a few microns away from the blood supply. Particle size definitely affects how fast a particle can diffuse. The smaller the particle, the faster it moves, and therefore, the quicker the concentration gradient is eliminated. Compare the size of glucose to the size of a globular protein. The comparison is analogous to the size of a mouse versus an elephant. In addition, some molecules require membrane channels to enter the cell channel-mediated diffusion. Temperature is important. We know this from everyday experience. If you want to dissolve sugar or salt in water quickly, you raise the temperature of the water. Living cells have certain limitations on this parameter because we maintain our body temperature within strict limits. We can't boil our cells to speed up diffusion. Gradient psi. Z refers to the difference between the areas with the highest and lowest concentration of a substance. The greater the difference, the steeper the concentration gradient and the faster diffusion proceeds. Lipid solubility affects how easily a solute can get through the plasma membrane. Because cell membranes are comprised predominantly of lipids, particles that are lipid soluble, such as dissolved gases, steroid hormones, and certain vitamins, can pass freely through the plasma membrane. The presence of protein channels, their size, Number and charge of the diffusing particle influence the rate of diffusion. Electrical forces can either attract or repel certain solutes. Many particles carry a charge when dissolved in water. As you know, opposites attract. Therefore, particles of opposite charge will be attracted to one another, whereas particles with the same charge will repel each other. As we will see later, the inside of the cell is negatively charged relative to the outside of the plasma membrane. The negative charge inside the cell pulls positively charged ions into the cell and repels negatively charged ones. Facilitated diffusion is a passive process. However, a membrane protein serves as a facilitator to help substances that are either too large or are not lipid-soluble get through the membrane. The facilitators are carrier or channel proteins. A good example of facilitated diffusion is the transport of nutrients the cell requires, such as plenty of glucose and amino acids. Glucose is too large to diffuse directly through the plasma membrane, and amino acids carry a negative charge and would be repelled by the negative interior of the cell. Therefore, carrier proteins or channel proteins are needed to get these critical nutrients into the cell. Facilitated diffusion using carrier proteins make use the transmembrane integral proteins. We shall see that when the appropriate type of molecule binds with its specific receptor, the protein carrier changes shape and moves the molecule into the cell across the plasma membrane. Glucose is critically important for cells. It is the favored substrate to use in the production of ATP, and as we know, ATP powers almost everything going on in the cell. The example above shows that glucose is more concentrated in the extracellular fluid than in the cytosol, but cannot diffuse directly through the plasma membrane because it requires a carrier protein. Although almost all cells move glucose across their membranes, some types of cells, such as fat and muscle, can only do this effectively when the hormone insulin is present. Inadequate insulin means that many cells can't take glucose out of the blood and transport it into the cell. Therefore, the blood levels of glucose following a meal get abnormally high. Since the glucose isn't being taken up by the cells, it remains in the blood, is filtered by the kidneys, and lost in the urine. Channels are also transmembrane proteins that form an aqueous pore to allow ions or water to pass through. The channels are selective in the sense that their openings or pores are of different sizes. Therefore, a channel with a small diameter pore can only let in small solutes. In addition, the amino acids lining the channel carry a negative charge, so the difference in charge between the solute and the channel protein will determine how well the solute can enter the cell. Another example of a special type of passive transport is osmosis, or the diffusion of water. As stated previously, water is chemically unique compared to other types of fluids. Because of its structure, water in body fluids maintains body temperature, provides protective cushioning, maintains cell shape, acts as a lubricant, reactant, solvent, and plays a key role in transport. All life and physiological processes on Earth have evolved around the unique properties of water. Water constitutes 60 to 90% of the total body mass of animals. The amount of water in the human body depends on the fat content of the individual. Lean individuals are 60 to 70% water. Heavy individuals are 40% water. If the average size individual has a total body water volume of 40 L, then 25 L or 40% of body weight is found in the intracellular fluid or cytosol. Interstitial fluid accounts for 12 L, and plasma makes up 3 L. Osmosis is the diffusion of water through a selectively permeable membrane that is freely permeable to water, but not freely permeable to solutes. Movement of water across a membrane is from the side with more water and therefore less solute to the side with less water more solute. In other words, Water moves down its concentration gradient. Water molecules flow towards solutions of higher solute concentration because such solutions have a lower water concentration or higher osmotic pressure. Osmotic pressure is the ability of a solution to cause osmosis of water. Another way to look at is that solutes suck. The solution with the greatest concentration of solutes will draw water to it. If there is a higher concentration of solutes, there must be a lower concentration of water on that side of the membrane. Water movement continues until water concentrations, and therefore solute concentrations, are the same on either side of the membrane. In the illustration above, equal volumes of aqueous solutions with different solute concentrations are separated by a membrane that is freely permeable to both solute and water. Both solute and water move down their respective concentration gradients. Equilibrium is reached when the water and solute concentrations on both sides of the membrane are the same. Notice that the volume of the solution stays the same on each side of the membrane because the membrane is permeable to both solute and water. Now consider the same solutions except that the membrane is impermeable to the solute. Water quickly diffuses from left to right and continues to do so until its concentration is the same on the two sides of the membrane. Equilibrium results from the movement of water along. But the movement of water leads to dramatic changes in the volumes of the two compartments. As water diffuses into the right chamber, the point is finally reached where the hydrostatic pressure, the back pressure exerted by water against the membrane within the chamber, is equal to its osmotic pressure, the tendency to resist further water entry. As a rule, the higher the amount of non-diffusible or non-penetrating solutes in a cell, the higher the osmotic pressure and the greater the hydrostatic pressure that must be developed to resist further net water entry. This situation mimics osmosis across selectively permeable membranes of living cells. However, major changes in hydrostatic and osmotic pressures do not occur in living animal cells because they lack rigid cell walls. Osmotic imbalances tend to swell or shrink cells due to net water gain or loss, until either the solute concentration is the same on both sides of the plasma membrane, or the membrane is stretched to its breaking point. The ability of a solution to cause osmosis movement of water through a membrane is measured as the osmotic pressure and expressed in mmHg. Osmotic pressure is the force with which water moves through a membrane into a solution with a higher solute concentration, lower water concentration. Osmotic pressure due to dissolved solutes is a negative pressure. Osmotic pressure sucks the water across the membrane. The force is measured by applying pressure to the top of a concentrated solution that prevents flow of water into it. The external force that is applied opposes osmotic pressure so that no net osmotic flow occurs. This force of pushing against a fluid is the hydrostatic pressure. Osmosis determines the distribution of water in the various fluid-containing compartments of the body cells. Interstitial fluid, blood. Osmosis continues until osmotic and hydrostatic pressures acting at the membrane are equal. Water is forced out of capillary blood by hydrostatic pressure of the blood against the capillary wall. But many solutes in the blood are too large to cross the capillary membrane, so they draw water back into the bloodstream. In the illustration above, blood entering the arterial end of a capillary has a pressure of about 35 millimeters Hg. Osmotic pressure, or the pressure caused by solutes, which opposes hydrostatic pressure, is 25 millimeters Hg. Thus, there is a net filtration pressure of 10 millimeters Hg. This is the pressure that squeezes fluids and small solutes out of the capillary and into the interstitial fluid where the tissue cells can select the materials they need. On the venous end of the capillary, hydrostatic pressure is low 17 millimeters Hg since fluid has been forced out of the capillary. but osmotic pressure hasn't changed. Therefore, at the venous end of a capillary bed, osmotic pressure exceeds hydrostatic pressure. Net filtration pressure is -8 millimeters Hg, and interstitial fluid containing cellular waste products is sucked into the capillary. This explains in part how nutrients are distributed to tissue cells and waste products are taken away. There are a couple more terms that we need to define in order to complete our discussion of how water and solute concentrations can affect living cells. The terms are osmolarity and tonicity. To begin, osmolarity and tonicity are not the same thing. Osmolarity or osmotic concentration is defined as the total concentration of solutes in an aqueous solution, or more specifically, the total number of individual atoms, ions, and molecules in the solution. How much water's concentration is decreased by solutes depends on the number, not the type of solute particle, because one molecule or ion of solute displaces one water molecule. For example, if a solution contained 100 glucose molecules and 100 sucrose molecules, its osmolarity is 200. Osmolarity in cells and other body fluids is measured in moles of solute/L of solvent. Tonicity refers to the ability of a solution to change the shape or tone of cells by altering their internal water volume tono equal tension. A solution's osmolarity is based solely on its total solute concentration. Its tonicity is based on how the solution affects cell volume, which depends on 1. The solute concentration, and 2. Whether or not the plasma membrane is permeable to the solute. Osmotic imbalances cause animal cells to swell or shrink due to net water gain or loss until the solute concentration is the same on both sides of the membrane, or the membrane is stretched to the breaking point. Solutions with the same concentrations of non-penetrating solutes as those found in cells, 0.9% saline or 5% glucose, are isotonic, the same tonicity. When a cell is placed in an isotonic solution, it retains its normal shape and exhibits no net loss or gain of water. For every water molecule that leaves the cell, one replaces it from the solution. Solutions that are more dilute contain a lower concentration of non-penetrating solutes than the cell's cytoplasm are called hypotonic. Cells placed in a hypotonic solution swell as water rushes into them. Distilled water is the most extreme example of hypotonicity. Because it contains no solutes, water continues to enter cells until they finally burst or lyse. Solutions with a higher concentration of non-penetrating solutes compared to the cytoplasm of the cell are hypertonic to the cell. Cells immersed in hypertonic solutions lose water and shrink. Cell shrinkage is called crenation. This picture compares the effect solutions of different tonicity have on red blood cells. Like all cells, the specific shape of RBCs is important for the cell to be able to carry out its special function. If the shape is altered, the cell cannot behave or function normally. Since RBCs carry oxygen, changing their shape can have detrimental effects system-wide. Notice how the hypotonic solution makes the RBC swell and even lose its biconcave structure. The swelling would make the plasma membrane weak, and the cell will lyse when it tries to squeeze through tiny capillaries. Placing a red blood cell in a hypertonic solution causes the cell to shrink, and its plasma membrane takes on a spiked appearance. With this shape change, it's likely that the RBCs would get stuck in capillaries and create clots. We've looked at what can happen to a single RBC as a result of tonicity changes. Now let's take a look at how tonicity changes can affect the body. Of all the major cations in body fluids, sodium has the most significant osmotic effect. Some of the major roles of sodium include nerve impulse conduction and muscle contraction, as well as regulating water movement between fluid compartments and affecting blood pressure. When sodium levels in blood plasma become elevated, the sodium diffuses into the interstitial fluid. The increased sodium concentration in the interstitial fluid causes water to flow out of the body's cells. Hypernatremia is the term for elevated sodium concentration in the plasma. What conditions lead to hypernatremia? We can use a marathon runner as an example. The runner loses water and sodium from the plasma by sweating. More water is lost than sodium, so the sodium concentration of the plasma increases. This causes the body's cells to shrink. Shrinkage of neurons results in dysfunction of the central nervous system, where sodium and potassium balance are especially critical. Therefore, a person suffering from hypernatremia may experience symptoms that include confusion, bizarre behavior, combativeness, hallucinations, seizures, lethargy, coma, and even death. Conversely, hyponatremia occurs if one becomes overhydrated with pure water without replacing electrolytes. This dilutes out the plasma. In this situation, water moves into cells, causing them to swell. Although life on Earth is based around water, a person can develop water toxicity. What happens in the case of our marathon runner is that the blood becomes too dilute. The runner loses both electrolytes and water from sweating, but only replaces the water component by drinking pure water. The body's cells are now saltier than the plasma and interstitial fluid, so water flows into the cells, causing them to swell. During the initial stages of water toxicity, hyponatremia, the person behaves as if they are drunk. Additional symptoms include fatigue, weakness, dizziness, headache, confusion, fainting, seizures, coma, renal failure, heart failure, liver cirrhosis, and even death. Because hyponatremia causes fluid to shift from the ECF compartment to the ICF, treatment entails administering diuretics and infusing a hypertonic salt solution. This causes a fluid shift from ICF to ECF, preventing further swelling or bursting of cells. Most people can't even fathom that too much water is dangerous. A case in point is the tragic death of a young woman. The following are a few excerpts from a newspaper article covering the tragedy. Jennifer Leah Strange, a 28-year-old mother of 3, was one of about 18 contestants who tried to win a Nintendo Wii gaming console by determining how much water they could drink without going to the bathroom. The show's DJs called the contest, "Hold Your Wii for a Wii." She commented on the tape that she looked pregnant, and a female DJ agreed. "Oh my gosh, look at that belly. That's full of water. Come on over, Jennifer, you okay?" The DJ asked. "You going to pass out right now? Too much water. They keep telling me that it's the water, that it will tell my head to hurt, and then it will make me puke," she said. Another DJ laughed. "Yeah, they signed releases, so we're not responsible. We're okay. And if they get to the point where they have to throw up, then they're going to throw up. And they're out of the contest before they die. So that's good, right?" Another one said. "Hey, Carter, is anybody dying in there?" a DJ asked during the show. "We got a guy who's just about to die," the other responded, and all the DJs laughed. The Sacramento County Sheriff's Department decided to pursue the investigation Wednesday after listening to the tape obtained by the Sacramento Bee newspaper. Sheriff spokesman Sergeant Tim Curran said, "The last passive process we will discuss is filtration. Filtration is the process that forces water and solutes through a membrane or capillary wall by fluid or hydrostatic pressure. Like diffusion, filtration is a passive transport process and involves a gradient. However, the gradient for filtration is a pressure gradient that pushes solute-containing fluid filtrate from a higher pressure area to a lower pressure area. As mentioned previously, in the body, hydrostatic pressure exerted by the blood. It forces fluid out of the capillaries, and this fluid contains solutes that are vital to the tissues. Filtration of blood by our kidneys is another example. A filtration membrane is formed from the glomerular capillaries and special cells called podocytes. Both the capillaries and the podocytes have different size pores or slits, which serve to retain blood cells and most proteins, but allow the passage of smaller solutes. It is filtration that is the first step in urine formation.